Hello, welcome to your ear articulate file. The ear is divided into the outer, middle, and inner ear. The outer and middle ear participate in hearing only. The inner ear functions for both hearing and equilibrium. I will expand on these topics throughout the lecture. The external ear consists of the auricle, or pinna, circled here, and the external auditory, or acoustic meatus. Most of our auricle is composed of the helix, which means rim. Inferiorly, the helix leads to the lobule. Superiorly, the helix terminates at the crus of the helix. The space superior to the crus of the helix is called the concha. This has no known function except to provide one with the opportunity to remove the shampoo that frequently accumulates here. There is also an antihelix and the crus of the antihelix. The anterior bump is called the tragus, and some people, ouch, choose to pierce this portion of their ear. Directly opposite from this tragus is the antitragus, found on the antihelix. The only purpose of the auricle is to gather and funnel, and thereby amplify, sound waves coming into the external acoustic meatus. Additionally, the way the sound waves bump over the helix and antihelix provides the brain with clues if the sound came from above or below. The external acoustic meatus leads to the external acoustic or auditory canal. Within this canal are hair, sebaceous glands, these release oils, and ceruminous glands. Ceruminous glands are modified apocrine sweat glands. Apocrine sweat glands do not respond to temperature, and these glands produce cerumen, or earwax. Earwax traps dust, repels insects, and provides hours of entertainment as we stick probe-like objects into our external ear for the sake of removing it, all the while risking a perforated eardrum. The tympanic membrane looks much like a flattened cone that slopes inferior medially, and this structure separates the middle ear from the outer ear. Sound waves travel through the air and cause the tympanic membrane to vibrate. These vibrations will in turn cause the middle ear bones, the ossicles, to vibrate. I will talk about these later. If you are not careful, the cotton swab you use to clear out the cerumen, or earwax, in your ear can perforate your eardrum. Additionally, if you scuba dive and cannot equalize the pressure in your middle ear, the increased pressure from the water around you may burst your eardrum. This happened to my sister, and she described salt water flowing into her ear and then into her throat. This is because of a structure called the pharyngeotympanic tube that ends in the back of the throat. I will discuss this structure more later. My sister's eardrum did heal, but there was some scar tissue, and this has caused her to have some hearing loss because the scar tissue does not allow the tympanic membrane to vibrate as easily. The most common cause for a perforated eardrum is from a middle ear infection that leads to pus buildup that ruptures the membrane. A slightly less well-known reason why someone could have a perforated eardrum is because a father of a 2-year-old allowed the 2-year-old to stick Q-tips in both his ears before trying to walk down the stairs, but the 2-year-old ended up slipping down them, and his fall jammed the Q-tips further into his ears. Fortunately for McLean and his father, the tympanic membrane did not burst in this case. The middle ear, or tympanic cavity, is a small, air-filled space inside the petrous part of the temporal bone. 2 small holes are found here, the oval window and the round window. The lateral boundary of the middle ear is the tympanic membrane. The medial boundary is a wall of bone that separates it from the inner ear. Superiorly, the middle ear arches upward as the epitympanic recess. Its superior boundary is the roof of the petrous portion of the temporal bone, which is so thin here that middle ear infections can spread to the overlying meninges and brain. the posterior wall of the middle ear opens into the mastoid antrum, a canal leading to the mastoid air cells in the mastoid process. Infections can spread from the middle ear to the mastoid air cells using the antrum as a passageway. The tympanic cavity is spanned by the three smallest bones in the body, the auditory ossicles, which transmit the vibrations of the eardrum across the cavity to a fluid in the inner ear. From lateral to medial, the auditory ossicles are the malleus or hammer, which looks like a club with a knob on top, the incus or anvil, which resembles a tooth with two roots, and the stapes, which looks like the stirrup of a saddle. Sound vibrations will hit the tympanic membrane and cause the malleus to vibrate because it is attached to the tympanic membrane, which then causes the incus to vibrate, which in turn causes the stapes to vibrate against the oval window. This will create pressure waves in the fluid within the inner ear. Excessive pressure waves will be dissipated out of the inner ear through the round window. I will discuss this in more detail later. The pharyngotympanic tube, formerly named the auditory tube or eustachian tube, links the middle ear to the pharynx. This normally flattened and closed tube can be opened briefly by swallowing or yawning so that the air pressure in the middle ear equalizes with the outside air pressure. This is important because the eardrum does not vibrate freely unless the pressure in both its surfaces is the same. Infection and inflammation of the middle ear, called otitis media, usually starts as a throat infection that spreads to the middle ear through the pharyngotympanic tube. More children than adults develop middle ear infections because their pharyngotympanic tube is shorter and enters the pharynx at a less acute angle. If they are bottle feeding and their parents lay them back too far during feeding, this invites milk to enter the middle ear and can lead to infections. Children with persistent middle ear infections have their tympanic membranes lanced and ear tubes inserted during a procedure called a myringotomy. This tube allows pus to drain through the external ear, and the tube will fall out on its own. Two tiny muscles are also found in the middle ear. The tensor tympani, which is innervated by cranial nerve 5, the trigeminal nerve, and the stapedius, which is innervated by cranial nerve 7. When loud sounds threaten to burst your tympanic membrane, from the ossicles vibrating so strongly, these muscles reflexively contract to limit the vibration of the ossicles, and also prevent damage to your hearing receptors, which I will discuss later. When discussing the inner ear, there are 3 bony labyrinths and 3 soft or membranous structures that are housed in them. The bony labyrinths, in addition to housing the soft membranous structures, are also filled with a fluid called perilymph. So, the perilymph surrounds the soft membranous structures. In the table above, you see listed the 3 hard bony structures, and next to them in the column in the right, you see the soft membranous structures housed by the bony rooms. Again, remember, these structures will be bathed in perilymph. The soft membranous structures also have a fluid contained within them called endolymph. I like to think of this setup as an aquarium. The aquarium is a hard structure that contains some water and some soft structures called fish. The fish also have water-like fluid found within them. The aquarium represents one of the 3 bony labyrinths. The water of the aquarium represents the perilymph. The fish represent the membranous structures, and the water within the fish represents the endolymph. I am now going to read through the table and tell you the bony and soft structures as well as the function of the soft structures. The semicircular canals contain the semicircular ducts, and these are important for rotational equilibrium. The vestibule contains the utricle and saccule, and these structures are important for static, linear equilibrium. Lastly, the cochlea contains the cochlear duct, and this structure is important for hearing. On the next few slides, I will give more detail on these structures. The vestibule is the central cavity of the bony labyrinth. It lies just medial to the middle ear, and the oval window is in its lateral bony wall. Suspended within its perilymph are the two egg-shaped parts of the membranous labyrinth, the utricle and the saccule. This means little sac. The utricle is continuous with the semicircular ducts, the saccule with the cochlear duct. The utricle and saccule each house a spot of sensory epithelium called a macula. Both the maculae of the utricle and the maculae of the saccule contain receptor cells that monitor the position of the head when the head is held still. This aspect of the sense of balance is called static equilibrium. These receptor cells also monitor straight line changes in the speed and the direction of head movements, that is, linear acceleration, but not rotation movements of the head. I like to think that I have an overactive utricle and saccule, as I always know when honey is speeding without even looking at the speedometer. These regions are innervated by the vestibular division of cranial nerve 8, called the vestibulocochlear nerve. Each macula is a patch of epithelium containing columnar supporting cells and scattered receptors called hair cells. The hair cells synapse with sensory fibers of the vestibular nerve, which is the vestibular division of the vestibular cochlear nerve. Each hair cell has many stereocilia and a true cilium protruding from its apex. The tips of these stiff hairs are embedded in an overlying otolithic membrane, which is actually a jelly-like disc that contains heavy crystals of calcium carbonate called otoliths, which means ear stones. How does this all work? When one holds the head in a tilted position, the heavy otolithic membrane pulls downward on the macula of the utricle, bending the receptor hairs and starting an action potential in the vestibular nerve to tell the brain the head is tilted. Then, when you elevate your head again, the otoliths in the saccule pull downward on the hairs, and this starts an action potential in the vestibular nerve to tell the brain that the head is in an untilted position. The semicircular canals house the semicircular ducts, and within the ducts are receptors for rotational acceleration of the head. Each canal and duct lies in one of the three planes in space. The anterior and posterior and lateral semicircular ducts all have a swelling called the membranous ampulla. Each ampulla houses a small crest called the crista ampullaris, or crest of the ampulla. The three cristae ampullaris contain receptor hair cells that measure rotational or angular acceleration. The hair cells project into a tall, jelly-like mass that resembles a pointed cap or cupula, which means little barrel. The basal, or bottom portions, of the hair cells synapse with fibers of the vestibular nerve. Because the three semicircular ducts lie in three different planes, each crista responds to head rotation in a different plane of space. When the head starts to rotate, the endolymph in the semicircular duct lags behind at first, pushing on the cupula and bending the hairs. As the hairs bend, the hair cells depolarize and change the pattern of action potentials carried by the vestibular nerve fibers of cranial nerve 8. The cochlea is a bony labyrinth, and within it is housed the cochlear duct. Cochlea means snail shell and is a spiraling chamber. The modiolus is the pillar bone around which the cochlea spirals. The cochlea is filled with perilymph. The above picture shows a transverse section through the cochlea, and you can see the three compartments. The middle compartment is the soft membranous structure called the scala media, or cochlear duct. The region above the cochlear duct is the scala vestibuli, and the region below it is the scala tympani. Both these regions are filled with perilymph. The scala vestibuli is continuous with the vestibule near the base of the cochlea where it abuts the oval window. The scala tympani ends at the round window at the base of the cochlea. The scala vestibuli and scala tympani are continuous with each other at the apex of the cochlea in a region called the helicotrema. The roof of the scala media is the vestibular membrane, and the bottom of the scala media is the basilar membrane. Within the scala media is endolymph and the organ of Corti, which will be described on the next slide. The basilar membrane supports the spiral organ of Corti, the receptor epithelium for hearing. It consists of rows of hair cells whose tips, called stereocilia, are embedded in a gel-like tectorial membrane. The base of the hair cells synapse with the sensory fibers of the cochlear nerve. When the endolymph of the scala media undulates, this causes the basilar membrane to undulate or move up and down in a wave-like fashion. And this bends the hair cells into the tectorial membrane and starts action potentials that then travel toward the brain through the cochlear division of cranial nerve 8. Let's go through the entire hearing pathway. First, sound waves are funneled into the external auditory meatus by the pinna. There, the sound waves travel through the external auditory canal and finally reach the tympanic membrane. The sound waves cause the tympanic membrane to vibrate. The vibration of the tympanic membrane causes the malleus to move or undulate, which causes the incus to move, which causes the stapes to pound on the oval window. The stapes' repeated pushing on the oval window means that now the sound waves have been transferred into pressure waves within the perilymph found in the scala vestibuli. The pressure waves travel through the perilymph of the scala vestibuli, round the helicotrema, and continue in the perilymph of the scala tympani. The pressure waves are dissipated through the round window. These pressure waves also cause the endolymph to undulate within the scala media, or cochlear duct. These vibrations cause the basilar membrane to vibrate, and this pushes the hair cells on the basilar membrane into the tectoral membrane. The hair cells bend, and this allows the hair cells to become activated and release neurotransmitter on the cochlear nerve fibers, which then carry the action potential to the thalamus, and the thalamus will then route the action potentials to the primary auditory cortex, found on the temporal lobe. Sounds like another delicious diagram question.